Hello, everyone. Thank you for joining us once again. Today, we have Sergio Martinez, and he will talk to us about abasic sites in DNA. Please, Sergio, start your presentation.
Okay. Thank you, Catalina, for the intro. And then thank you to Melike and the ISCB Student Council for inviting me today. It's a great opportunity to share my work. So I'm going to share my screen. Okay, so can you see the slides? Can you hear me okay?
I can see them and I can hear you. I hope everyone else can too.
Awesome. Okay, thanks a lot. All right, so yes, Catalina was saying, today I'm going to be talking about several methods that we developed in our group to be able to map abasic sites, but also other forms of DNA damage and epigenetic modifications at single nucleotide base resolution. And some of this work has been done using human, several parasites, and some cancer models as our reference tissues. Okay, so I'm going to give you a little bit of background of who I am, and then just going to briefly introduce you the research focus of my work. And then finally, I'm going to give you a brief intro on the different methods that are available to be able to detect damage and chemical changes in DNA. And then I'm going to dive into the abasic sites mapping project. And briefly, at the end, I will just highlight in a slide just some of the other things that I've been doing related to this topic. And hopefully we can talk about it in later presentations. Okay, so I did a master's in chemistry in South Spain. And then went to do a PhD in bioinformatics at the EBI and finished it in 2015. And there I was working mainly in developing structural bioinformatics and cheminformatics approaches to understand how enzymes evolved and how the chemistry of enzymatic reactions change during evolution. And then since 2015, I've been a postdoc at the University of Cambridge and at the Cancer Research UK, where within the Bala-Subramanian group, which I'll introduce in a minute, we've been developing different types of methods to look at chemical modifications and secondary structures in DNA and RNA. Okay. So this is the Bala-Subramanian group. We're a very multidisciplinary group composed mainly of chemists, biologists, but also computational biologists, more informaticians. And we're based both at the Department of Chemistry in Cambridge downtown, but also in the biomedical campus in the south of Cambridge at the Cancer Research UK. The research focus of our group is twofold. So on the one hand, so on the left-hand side, you can see that we are interested in looking at chemical modifications that happen to the nucleotide bases. So several of you will be familiar about the mutational processes where you have C to T mutations, but there are also chemical transformations happening to cytosine. So perhaps the most common one is this methylation that happens in the 5th position of cytosine to produce what is known as the 5th canonical base, right? 5-MC. But there are, in DNA, there are about 15 to 20 different types of modifications, not only in cytosines, but also in thymine, in adenine, and also guanine. And then in RNA, it's even more. I think last time I looked at, there were more than 170 different types of modifications happening in the nucleotides present in RNA, which is very exciting. So the other part of our lab, we're interested in looking at secondary structures in DNA and RNA. And in particular, we look at type of secondary structure known as a G quadruplex, where it forms a 3D structure when you have repeats of guanines locally in the genome. So, for example, GGG, a triplet of guanines followed by a loop, and then another triplet of guanines, GGG, and then other loop, and then another triplet. Then that tends to form a 3D structure shown here. And then here is coordinated by a metal in the middle. And we're trying to investigate what is the function. So why do they appear, how they are regulated. Okay. So as you know, DNA is just not about the primary sequence, but you got other orders of complexity. So you have things happening at the DNA modification level. So for example, DNA methylation. But then you also have modifications happens in the histones. And then on top of that, then you also have that, histones and chromatin wraps around in a particular 3D structure that allows, the DNA to be very compactly present within the nucleus of a cell. Okay? So the questions that I've been interested over the last 5 years is, so do modifications exist? These chemical modifications on secondary structures, where are they located? Can we map them? How are they produced? Are they produced by enzymatic processes, or are they produced by oxidative damage or other processes? What's their function? What's their role in development? And how about in disease, in particular in cancer? Then more from a structural point of view, we are interested in finding out whether they alter the structure of DNA and chromatin, and whether they affect the binding of transcription factors to promoters, or polymerase activity, or gene expression. Okay, so DNA modifications. Here I'm showing you some examples. So you have, for example, this modification called 5-HMU, 5-hydroxymethyluracil, that is very well described to be present in parasites. So in Leishmania and trypanosoma. So parasites that are common in some, for example, in the sleeping sickness and some other parasite diseases. And then we have little information on whether they are also present in mammals. Okay. So 5-methylcytosine, they are present in mammals and bacterial organisms. And then modifications such as N6-methyl-A, they have been very well described in bacteria and nematodes, but we have some evidence that they might be present in mammals, but not very clear yet. Okay, so this is just some examples of some of the modifications that we're looking into. So here I'm showing you the Watson and Crick hydrogen bonding. And here in circles, I'm showing you the hotspots of modifications happening in DNA. So you have this position in cytosine. Okay, you tend to have a lot of modifications there. Similarly, in thymine, there are modifications happening there, and then in the methyl group here. In adenine, so we talked about this N6-methyl-A, but the majority of modifications happen there. However, in guanine, the majority of modifications happen here in this nitrogen nitrogen 8 and this nitrogen 7 and then this carbon 8. Okay. So it's well described that nucleotide modifications are produced by families of enzymes. So here I've color-coded the enzymes involved in these pathways, both for the cytosine modification pathway at the top and the thymine modification pathway at the bottom. So you can see that you have methyl transferases, so enzymes that include, catalyze the incorporation of a methyl within the nucleotide, and then you have enzymes that catalyze oxidation of that methyl group, and then you have what are known as excision enzymes, so enzymes that detect the modification, they excise it, so they break the glycosidic bond, they excise it, and they form an abasic site, okay? And then there is a machinery in the cell called the base excision repair that takes that abasic site back to either the cytosine or the thymine, okay? So you have also a family of enzymes that, if you like, they go across pathways. So these are known as the deaminases. So for example, the ApoBec or AID that, for example, they catalyze the conversion of 5-methylcytosine into thymine, okay? And then you have other more exotic modifications, for example, this JGT, where it takes this modification and then it takes a glucose molecule, so a sugar molecule, and then it adds the sugar molecule there. So, you have sugars floating, covalently linked to DNA, which is quite exciting, I would say. Yeah. So that modification in particular is called base J, and it's very common in parasites. Okay, so as you can see, these abasic sites, named here as AP, they play a central role in the mechanisms of excision and repair of modified bases. So just briefly, in the next few minutes, I'm going to tell you about the different methods that are available to map modified bases. So the first one is mass spectrometry. So very simply, so you isolate DNA from a cell, you digest it into the individual nucleotides, and then you run that through a liquid chromatogram followed by mass spectrometry, tandem mass spectrometry. And then you can detect, using standards, you can detect the modifications that are present within your DNA. Okay, so for example, here Y, we've been able to detect here in the MS chromatogram. Okay, and this method is commonly used for determining the global levels of modification, and it's a very highly sensitive method, and it usually used to be able to discover new modifications. However, one of the limitations of MS is that it doesn't allow you to know exactly in the genome where the newly discovered modified base, where does it happen exactly, okay? So in order to do that, then you have to do more enrichment strategies followed by sequencing, okay? So here, some of you might be familiar with ChIP-seq and these approaches. So here, this works similarly. So you have DNA that is modified, you fragment it, and then you have an antibody or maybe a chemical enrichment strategy that is able to isolate the reads that have the modification, and then you do sequencing and align to a reference. And then by do big calling or single nucleotide base calling, if you like, you can map modified bases genome-wide at, in some cases, a nucleotide base resolution. So delving a bit more into the sequencing strategies, they're classified based on their resolution. So you've got single nucleotide methods where you for example, you have chemical conversion methods that rely on this differential base pairing. So some of you might have heard about bisulfite sequencing. And what you do there is that when you treat DNA with bisulfite, then cytosines, 4' cytosines and carboxycytosine, they get converted into uracil, therefore they are read as Ts. However, these modifications, MC and HMC, they remain as they are, as MC and HMC, and therefore they are read as Ts. So therefore, by you can use this method to be able to differentiate things that have been converted by bisulfite compared to things that haven't been converted. There are other methods that rely on, if you like, stalling of DNA polymerase. So sometimes you have bulky modifications that they make the polymerase to stall at that position. And then by looking at pile-ups and this asymmetric distribution of aligned reads, then you can also detect these modifications. And more recently, there are methods, for example, such as PacBio and also nanopore methods, where if you have a modification, when it goes through the pore, for example, in nanopore, then you can detect this differential potential and therefore being able to detect it at single nucleotide base resolution. And more low resolution methods, you have chemical enrichment strategies by using antibodies or chemical enrichments or pull down strategies. Okay. All right, so briefly, I'm going to tell you one of the projects that I've been working in the last year or so. We just got published end of last year. This was work with some of our collaborators here at the chemistry department, Jane and Fumi. And we've come up with a method to map abasic sites, single base resolution. So just following up from the intro, DNA damage and abasic sites are a form of DNA damage. And as you know, DNA damage are part of the hallmarks of cancer and aging, whereby, it is hypothesized that by having the increases in DNA damage within the cell, then you're more likely to suffer from cancer or to age faster. Okay, so in a way, the cell, when the DNA of the cell is damaged, the cell is subjected to a dilemma, if you like. On the one hand, you have the repair mechanisms that fix that modification. That, for example, when you have repair of ADOXG, which is a very common form of DNA damage through an abasic site intermediate. But in some other cases, some of these modifications lead to mutagenesis that in some aspects might be beneficial for the cell to have that modification to perhaps have a competitive advantage. So, the cell is in a dilemma sometimes on whether to repair it or just leave it on and let mutagenesis to happen. So in this paper by Anna Potash, there were some of the methods that have been described to map abasic sites. So this is our method here, a single nucleotide abasic site. And just in parallel, there was a method called abasic site, APSIC, which it's an alternative to our strategy to be able to also detect abasic sites. But what is an abasic site? So essentially, you've got that your nucleotide, so your base here is missing. Okay, so that's about it. We know that there are around 10,000 abasic sites per cell per day, and then they are products of DNA damage. And then, as I showed you before, they are key intermediates of the pathways of repair and excision. So briefly, how do we detect them experimentally? So here I'm showing you our reads or DNA containing basic sites. So Jane and Fumi, they were able to develop a chemistry to label the abasic sites using a tag, and then to be able to, upon processing and library preparation, they were able to essentially degrade the abasic site, the DNA to the position where the abasic site is located. So that at the end of the day, you end up with something like this, right? You have enriched for the reads that contain the abasic site, and then on top of that, then you have that upon sequencing, and then you do pair N and read one, beginning of read one, then that's what is marking the location of the abasic site. So yeah, I mean, for those of you that are like chemistry, so basically the key of this method is that the abasic site goes through an open form here, and then you can detect that with chemistry that binds to the aldehyde, and then you can pull down abasic sites. So roughly, bioinformatically, you end up with something like this. So you have these pile-ups, okay? And so, for example, here, these are pile-ups of read ones, okay? And then you are interested here on looking at these differences of start of alignment. So you have 9 alignments here in the treatment, and then 1 alignment starting the control. So you're interested in looking at this difference and see whether the difference is statistically significant, and therefore to be able to call it as an abasic site. Okay, and similarly for the reverse strand. So the processing works. You end up with essentially count data, right? So you have your alignments, you count alignment start sites, and then you normalize by library size. And then you can do the modeling and scoring that is very commonly known for other count statistics such as RNA-seq or ChIP-seq. So briefly, you fit a negative binomial distribution to the count data, and you use it for estimating the variance. And then you do that in a form of the GLM, so a generalized linear model, with the treatment and the control counts. And then you're scoring, you do significant tests under the null hypothesis there is no difference between the treatment and the control. And then you do multiple testing correction, volcano plots, and thresholding based on the statistical significance. So you end up with volcano plots like this one, where you use these thresholds to call a basic site. So here are positions, so positions in the genome, where you've done these tests, and then you can set your thresholds, and then you call abasic sites based on that. And finally, the things that takes most of the time, because we do a lot of collaboration with our chemists and biologists in the lab, then it's about really understanding what is the function of these maps, these abasic sites. So we look at profiles, we look at genomic enrichment analysis motifs, and then we do many different types of correlations against RNA-seq experiments or ChIP-seq experiments, etc. So here I'm showing you an example. So here there's an enriched region in a parasite genome where we were able to detect a lot of abasic sites. And then you can see that here in the genome browser that you get these pile-ups that I was mentioning earlier, where there are a lot of reads that start the alignment, and then you can't see that in the input, in the control. And therefore, we use this as a proxy to be able to say, all right, these are the basic sites. So in that particular genome, then we were able to determine that the majority of abasic sites that were present in thymine, okay? And as I said, just like in mammals, we have a lot of modification happening in cytosine with 5-MC. In parasites, the majority of epigenetics happens in thymine, right? Which is quite different, given that we are all eukaryotes at the end of the day, right? So yeah, parasites like modifications in thymine, and then we use this strategy to be able to map abasic sites in the genome, in their genome. Then we also map them in the human genome, and here I'm showing you some of our results. So we've been knocking out the enzyme, so the base excision repair enzyme responsible from repair of abasic sites. And then we've been able to determine whether they are more present in promoters or in 5' UTIs. And we could see that upon knockdown with AB1, then we could see an enrichment of abasic sites in functional regions like UTIs and promoters, and not so much in introns and intergenic regions. So one of the things that we see that there is a potential application for this method is when it comes to understanding more about toxins. So this was a paper that was published earlier this year, where they described a mechanism whereby colibactin, which is a common bacterial toxin, binds to DNA, and then it methylates it, well, it alkylates it, and then it generates genotoxicity, mutagenesis, and therefore colon cancer as a result. So here, little is known about the molecular intermediates of this process, and then the potential application of our method is to be able to hypothesize here whether that intermediate might be an abasic site, the intermediate to genotoxicity, and yeah, that's something that we're currently looking at. So just briefly, my last slide. There are other things that we're looking beyond just looking at abasic sites. So we're also interested in looking at oxidation of cytosine in glioblastoma. So we've been able to see that there is a decrease of this oxidation in the tumor compared to the margin. And then we're exploring where we can use this as a biomarker for the development of glioblastoma. And then other groups are also being able to identify that loss of 5-HMC is also observed in liver cancer and kidney cancer. And so we're investigating whether this is a common mechanism of cancer for cancer to develop. So there is another study that was published a couple of years ago, where we were studying the link between the positioning of this modified base, 4-methylcytosine, and nucleosomes, in particular the lysins of nucleosomes. And then we were able to find an overlap significant overlap between this 4-methylcytosine and the positioning of nucleosomes. And then using biophysical methods, we're hypothesizing that perhaps there is some covalent binding between the nucleosome, so the lysis in the nucleosome, and the DNA, so the modification in DNA, something that we're investigating further. And then work in progress at the moment is some of our parasite work, where we are mapping the modification base J that I was showing in the first slide, and also other forms of thymine modifications to try to understand more about how parasites, in particular Trypanosoma brucei, how they do the infection of the human cells. Okay, so that's it. So yeah, just acknowledgements. So obviously the Balasubramanian group, this is a fantastic group to be part. So, if you're looking for doing a postdoc or a PhD, please, get in touch. It will be, it will be great to have you on board. And everyone at the Cancer Research UK. And yeah, obviously the ISCB Student Council for inviting me to speak today. And if you have any other question, just drop me an email. And I'm happy to follow up any of the discussions and take on any questions now. Thanks.
Thank you so much, Sergio, for this very interesting talk. Now we can wait for questions. There is a small delay between the transmission on YouTube, so yeah, we have to wait a few seconds to see if there are any questions. Meanwhile, I want to remember everyone to follow us on Twitter and Facebook, so you can know about the next talks and the next abstract submission, so you can participate with your own talks on these webinars. Okay, there is a question.
Yeah.
Do you know if there are certain tissues or cell types that are more prone to AP sites?
Yeah, good question. We don't know. We don't know. At the moment, that's something that needs to be explored, actually. I mean, we just came up with the method and sort of a systematic study of abasic sites in different tissues or cell types. That's something that it needs to be done, really.
I also have a question. I think you you answered it a bit, but it's if the modifications on nucleotides can have an effect on how the nucleotides bind between them and yeah, in the DNA structure, and make the sequence more prone to damage or mutations.
Yeah, so yeah, it has been described that having modifications such as formyl-C, it has an impact on the structure, and there have been some crystal structures of DNA with a formyl-C, where you can see that the DNA has doesn't look like bDNA, which is the one that we're familiar. So definitely, when you have a formyl-C, you have an alteration of the structure. Having said that, there are also some other publications that suggest that that's not the case. This is essentially work from the last couple of years. So yeah, there are some groups that say yes, there is alteration of the structure. Some groups say no. So yeah, that's what the field is at the moment regarding formyl-C. Yeah.
Thank you. I think there are no more questions, so thank you again for your talk. Thank you to the people that joined this webinar, and goodbye.
Thanks a lot. Bye-bye.
Bye.